The next talk is by Barry-Reidy. Yeah, and she's going to talk about conserved DNA elements that may influence expression of reproduction. The floor is yours.
Okay, so first of all, I'd like to thank you all for coming along today. I'm aware we're very close to the end. We're all looking forward to go home, so I'll try not to keep you for too much longer. So I'm going to be speaking today about these bovine beta-defensins. So first of all, what is a bovine beta-defensin? So as the name defensin would suggest, these molecules were first discovered as being part of a wider class of molecules known as host defense peptides. And these were shown to have antimicrobial and a wide range of immunomodulatory functions. However, more recently, selected members of this gene family have been shown to be involved in reproduction. So just to give a couple of examples, probably the most famous is beta-defensin 126. It has been shown in a number of species that this gene is expressed by the epithelial cells that line the epididymis, and from there they are secreted and they attach to the surface of spermatozoa as they pass through the tract. So what you're seeing in the image here is an image taken by somebody else in our group, and this is a mature bull ejaculate, and the green colouring here is the beta-defensin peptide, indicating that this is found on the tail region and on part of the head region of the mature bull spermatozoa. To give another example, this same gene, when people looked at this in the human, they found a coding region deletion that actually renders males subfertile. Another example again, when somebody has partially deleted this gene locus in the mouse, they found that males were completely infertile. So it does seem that at least some of these genes are involved in reproduction. So to go back to the bovine, we believe that there are up to 57 protein coding genes in the bovine genome, and these are organised across 4 chromosomes. Our group is particularly interested in a subset found on chromosome 13, and we are interested in these because we found that they are expressed only in the sexually mature bull. So they're not expressed in the sexually immature bull, and they're not expressed in the female. Now that would absolutely suggest that factors such as androgens are regulating the transcription of these genes. However, across all species, very little has actually been shown empirically as to how these genes are regulated. So to go back to some more basic principles before I get into my methods, transcription of genes is regulated by proteins called transcription factors. So if you imagine that this is the first exon of a protein coding gene, just upstream or 5' of that, you've got your gene promoter region. And these boxes are representing graphically transcription factor binding sites to which these nuclear proteins called transcription factors are able to bind. And in doing so, they provide a 3D complex for transcription to be initiated. So over time, through experiments like CHIP and ChIP-seq, huge libraries have been built up containing the short DNA motifs that we now know various transcription factors are able to bind to. And that allows people like me to search gene regions of interest for potential transcription factor binding sites. So while, of course, the promoter region is very important in regulating transcription of a given gene, represented here, genes are also regulated by more distal regulatory elements, and these so-called suppressor or enhancer elements can regulate one or a number of genes in tandem, and that's very relevant when you consider a situation like the beta-defensins, where we've got a family of genes located beside one another in a gene locus. So my objective then was to predict some of these molecular mechanisms that regulate expression, particularly of these reproduction-associated bovine beta-defensin genes. And I've used an in silico approach towards that. And what I've been trying to do is to predict in the non-coding genome elements which might have gene regulatory function. So this is a hypothesis generating exercise towards directing our experimental work, because we were starting really from nothing in terms of what might be regulating these. And what I'm aiming to do is to identify conserved non-coding genomic regions. So what does that mean? If you take the same sequence of DNA between 2 mammalian species, let's imagine we've got the bovine and we've got the human. If your blue box here is indicating a protein coding region and the clear is non-coding, in protein coding regions, generally because there's a selective pressure, we see quite a high level of conservation. And when people search for SNPs, they're identifying localized regions of genetic difference on that background of quite high similarity. If you look at the non-coding genome, we're actually looking at more or less the reverse situation. Because this is mostly non-functional, we are seeing quite a high level of sequence dissimilarity. And what I'm trying to do with this approach is to identify within that areas that are conserved. Now, these are not conserved because they are protein coding, we know that. I'm hypothesizing they are conserved between mammalian species that have been separated by millions of years of evolution because they are functional in some way, and perhaps because they function by binding important transcription factors. So I have done something called transcription factor overrepresentation analysis. Now, what is that? Basically, I've used publicly available software called Opossum to statistically analyze the rate of occurrence of a given transcription factor binding site between two sets of gene promoters. So what are my two sets? So on one hand, I've taken the promoter of a given beta-defensin from each of a number of different species. So let's imagine I've got bovine beta-defensin 126, human, mouse, so on, so on. These blue boxes are indicating the instance of an occurrence of a transcription factor binding site, and I'm statistically comparing that rate of occurrence to random chance, essentially, and that's modelled by randomly selected genes. And I've got quite a high pool of random genes versus my defensin genes, so that's basically modelling random chance, because when you've randomly selected genes, you're expecting that there's not any commonality among that pool in terms of how they are regulated. So using this approach then, I've assessed 290 transcription factors, all within the 5,000 base pair promoter of each of 15 beta-defensin genes. And for each of these 15 genes, I've looked at between 7 and 14 mammals, depending on how many genomes a given gene is actually annotated in. So to give a brief overview of the results, this software imposes two different statistical measures, and there are cutoffs recommended above which you're looking at transcription factors that are overrepresented. So to start from that initial pool of 290, we were able to boil that down for each gene to between 3 and 7 different transcription factors. So this method has been quite rigorous in reducing your pool of initial candidates right down. And what we're seeing, what I've indicated in bold here, are transcription factors that when we go to the literature are known to be associated with the male reproductive tract. So of course we've got androgen receptor, we've also got glucocorticoid receptor, retinoic acid is the vitamin A receptor, and so on, so on. So for all of these transcription factors in bold, there is evidence to suggest that they regulate the expression of genes in the male reproductive tract. So I'm going to talk about one example today, just in terms of time. I'm going to mention beta-defensin 126, since we touched on this earlier on. So what is this looking plot? So I've plotted the two statistical measures, the Z-score versus the Fisher score, against one another. Each of these individual points is an individual transcription factor, remembering that we started with the pool of 290. These two lines that you're seeing here are the recommended statistical cutoffs, or the applied thresholds for each of the two scores. And then in your upper quadrant, what you're left with is that small pool of transcription factors which are found to be statistically overrepresented in those mammalian promoters relative to your random chance background. So what we see here for this gene, and I'll explain in a second why this is interesting, we've got this AP2 family coming up, so several different members of the AP2 family, probably because they bind to quite a similar DNA motif. And to represent this data a different way, what we're looking at now is that 5,000 base pairs of promoter region for each of the species represented graphically. So you've got each of your species, and then you've got your 5,000 base pairs. If you imagine your transcription or translation start site begins here on the right, you've got 1,000, 2,000, all the way up to 5,000 base pairs upstream. And what's interesting about this, again, is that we've got this conserved AP2 family binding site, conserved across all the species that we see here, and conserved in a location quite close to the transcription start site. So we would posit that this transcription factor is very important in regulating the expression of this gene. And we knew nothing about this to start, but however, when we went to go to the literature, we were very interested to find a paper that looked at ChIP-seq of genes that are expressed in the murine epididymis, and what they found was that genes that are responsive to androgen are regulated not only by androgen receptor, but also by this AP2 alpha, which is kind of acting as an accessory receptor, so is teaming up with androgen receptor to regulate genes in the epididymis, as separate from how androgen regulates genes in other organs in the body. So these mechanisms are very much tissue specific. So that very much correlated with what we knew about where this gene is expressed. So we were very interested in that. So I did talk earlier on about how when you're looking for regulatory mechanisms such as this, it's important not just to look at the gene promoter region. So the approach that I have just described is just looking at your 5,000 base pair promoter, but of course it's also important to look at potential elements. So we've looked at the promoter, and now we're going to look at what may or may not exist in a regulatory fashion within the wider beta-defensin locus, because again, you've got up to 19 genes sitting within one gene cluster on chromosome 13. So this is quite a complicated figure, but I am going to talk you through it. So what I did here was basically an alignment of that entire 300 kilobase region between the bovine and a smaller number of other mammals. So you can see you've got your pairwise alignments here on the left-hand side. And each of these black dots is basically representing the percentage conservation, percentage indicated on the side here. The percentage conservation at a particular location in this gene locus, as indicated on the bottom there. And what was interesting to us was that, of course, we were seeing protein coding in promoter regions quite highly conserved, but we were also seeing intronic regions and also intergenic regions conserved as well. So we were seeing things that are possibly important that were missed out with our initial approach. And what was very interesting to us was when we applied a recommended increased conservation cutoff, we see within this whole 300,000 base pairs, only 3 regions meeting this enhanced cutoff. And this enhanced cutoff is recommended because it's supposed to identify regions which are even more likely, again, to actually be functional. So I'm taking this figure is just an excerpt from a much wider figure, and I'm showing it because, again, it relates to beta-defensin 126. It's, relatively speaking, just downstream of that. So when we got this, we were of course interested to see what transcription factors might actually bind within this region. And we were very interested to see that we were coming up with things that very much correlate with what I talked about earlier on. So in this separate element, again, we're seeing transcription factor binding sites for androgen, so mediating the effects of, I suppose, sexual maturity in the male, and also this AP2 alpha. So to reiterate again, evidence from other species does indicate that in the epididymis, where we know this gene is expressed, these 2 factors are collaborating together to drive expression. So the results. So this approach certainly seems to be it has generated a lot of very logical hypotheses for us. So to start from quite a wide pool of possible factors, all these kind of scenarios that might regulate defensins, we have come out of this with a number of hypotheses that actually make a lot of sense for us in terms of directing our future wet lab work. So to talk about our most interesting hypothesis, we've got your epididymal epithelial cells where we know defensins are expressed. We've identified this AP2 alpha binding site in the promoter, the proximal promoter, and we hypothesized that when this gene is transcribed, it's interacting with this more downstream element where AP2 alpha is acting as a docking site to mediate the effects of androgen on gene expression in the murine epididymis. So again, this has come up with a lot of possibilities for us in where we would like to go with our future work. And you would wonder whether genetic variation in these so-called conserved regions may be contributing to bull fertility. So I'd like to take this opportunity to thank both my supervisors, Cliona and Kieran from Trinity and Teagasc. I'd like to acknowledge Dr. Karsten Hokamp, who's provided some bioinformatics support. I'd like to acknowledge the Irish Department of Agriculture for funding this work. Teagasc Wall Fellowship has funded myself personally, and I'd like to thank you all for listening. So I'd be happy to take any questions.
Thank you. Any questions? I think you need me to help me a little bit.
Sure.
As a simple animal breeder, there's a lot of language in there where I struggle. Can you tell me, you do the comparison between the species?
Yes.
How does that help you, or what answer does that provide? And the second question is, how do you conclude that these elements are important for fertility?
So the first question, I suppose, we're hypothesizing that if you were to compare bioinformatically a sequence of bovine DNA with the stretch of the same sequence in the human, these have been separated by millions of years of evolution. And obviously, you see, regions that are functional in some way are under much greater selective pressure and tend to deviate much less in sequence than areas that are non-functional. So this has identified non-coding regions which, for some reason, are conserved. Yeah. Now, we're hypothesizing that maybe because they act as binding sites.
Yeah, and you call them non-coding, but it doesn't mean they are non-functional.
It doesn't, absolutely.
In
So
fact,
they
you
don't
imply
code
they
for
are functional.
proteins. They could act as sites for epigenetic modifications. We don't know that they code for proteins, but we can only hypothesize as to what they may do outside of that.
Okay. And the regions you look are expressed in tissues that you consider important for reproductive performance, or is that?
Yes, so the interesting thing about these particular defensins is that they're expressed in the male reproductive tract, and as spermatozoa mature, they're moving through the epididymis, and the proteins that are actually secreted by the epididymis are very important for maturing the sperm, and we think beta-defensin 126 is one of these. So we're very interested particularly to know in what scenario are these defensins expressed.
Yeah.
With an idea towards tying that up with functional work that other people in our group are obviously doing.
Okay, so on the basis of this, you have somewhere where you can zoom in with your wet work in the lab.
Exactly.
And start looking on the,
Yeah.
instead of, okay.
Yeah.
Thanks very much. Any other questions? Jenny? You can use this microphone. Oh. She comes running to you.
I was just wondering whether you think folding of DNA is important, especially as you look at promoters or enhancers that are further away.
Absolutely. I mean, there's a lot of, and some of these elements may actually not be acting as transcription factor binding sites. They may actually be modulating that kind of folding, which certainly at the 3' UTR is modulating how genes are actually processed post-transcriptionally. So, I mean, absolutely, this kind of approach is considering one aspect of transcriptional regulation, but of course there are lots of other mechanisms that are very important as well.
Okay, thank you very much.